Okay, so I just have a quick plug here because I wasn't around during the morning sessions, and I understand there was an education workshop and a session. So if people are interested in education, I thought they might be interested in this initiative we just launched, where we're trying to develop do-it-yourself affordable lab equipment using microcontroller platforms like Arduino. I'm very happy to talk about this. If people are interested, I just want to put that up. So when it comes to my talk, I understand this is a sort of a black sheep session of biotechnology, and I guess this is the black sheep talk. I come from a biochemistry background, and probably there's too much biochemistry in this talk for many of you. So I'm going to skim through lots of stuff, but I'm very happy to talk and give detailed information about bits afterwards. So I like to talk about metabolism, and if you open up a textbook and look at metabolism, these kinds of diagrams is what you'll find. So the description of, or the sort of rationalization of metabolism of a cell is that it consists of different biochemical pathways, and these pathways allow the cell to sort of rebuild itself and gain its energy for division. Now, while that is sort of useful depiction of metabolism, it falls short actually to explain a variety of observations that we are making about metabolism. One of them is that actually the cell is not an orderly sort of a self-sufficient biochemical engine like this figure suggests. Instead, there is a lot of excretions from the cell and seeming inefficiencies in the way the biochemistry is dealt with. So we like to understand why the cell excretes so much stuff out, and also is sometimes taking in things that are supposedly essential for cell division. So if this was a sort of a system that has evolved to just provide energy to the cell, you will have expected that it should be a self-sufficient, self-closed system. So we can't really explain this in this sort of simple biochemical pathway conversion way. Another component that is not captured by the pathway diagrams like this one is that they tend to think about metabolism as isolated biochemical units. You know, A goes to C, and then another element is converted to F, and then F and C can combine to do something else. And you get these nice diagrams, but the reality is that everything seems to be connected to everything else. So this is just a small part of those chemical reactions I was showing earlier. And even here, we get a lot of cycling material, we get a lot of connections among different parts, and so on and so forth. And this is important because some of the stuff that I'm showing here is actually essential units to make up a new cell or set of proteins. So these are amino acids, for example. They're connected to each other, and they're connected to different sugars that the cell takes up. So we're increasingly thinking that this pathway view is maybe a nice way of rationalizing things, but it doesn't really help us to predict or understand or engineer cells' metabolism. So we've been working on, not just us, but many people have been thinking about or finding ways to think about differently about metabolism. In one way, a simplified way of thinking about this is as an electrical flow system. So what actually the cell is doing is taking substrates, mainly sugars, that are highly rich in electrons, and actually transfer those electrons through a bunch of redox reactions, and finally dump those electrons to what we call terminal strong electron acceptors. This is usually oxygen for many of cell types, but actually if you look at single-celled organisms like microbes or algae or cyanobacteria, they can use a variety of strong electron acceptors like nitrate, sulfate, and things like that. So this process of electron dumping is generally referred to as respiration, and the taking up of the sugars is known as substrate uptake. So we can start to think about a cell carrying up an electron flow process. And if we think about this process, then we can immediately see that there might be some limitations, and there might be some solutions that evolved over time to overcome those limitations. So I'm going to go through quickly some of those limitations that we also have encountered in our own research, and I'm going to talk about some consequences of this way of thinking about metabolism for engineering metabolism. So one of the simplest limitations you can think about is actually a resource allocation problem. So we are talking about an electron flow system that has to sort of take place over the cell, and you have a limited capacity to run these reactions because most of them, if not all, are catalyzed by enzymes, which you also have to produce. So there's an idea about resource allocation limitation, both in terms of the enzymes that you use and in terms of the space you have available. And note, please, that these processes, some of them are taking place over the membrane. Okay? So if there are now limitations on one aspect of this process, and I'm trying to flow electrons through the system, then there must be a solution. And usually the solution can be actually these overflow of metabolites from the cell that I referred to at the beginning. So we can actually, with this way of thinking, easily understand why some overflows are happening. And this is experimentally well observed. These are actually diagrams of metabolite flows within yeast cells. And you can see that some yeast cells have overflow into organic acids, including ethanol and acetate, which people produce in biotechnology, and some don't. And actually, the distinction is not clearly understood why some cells go into overflow and some don't, but we think that it is related to these electron flow limitations, in particular, perhaps, to the limitation of redox agents. The same phenomena, just as a side note, is also well documented in cancer cells, for example. So this switching between when the overflow happens and when not can also happen during the lifetime of a cell under different conditions. So we want to understand this through a modeling approach, how this overflow can arise through the resource allocation problem. So we developed a flux balance analysis model where we impose global trade-offs in the resources available to the cell in form of uptake and respiration mechanisms. And then we sort of force the cells to evolve and choose the optimal strategy in their environment. So in this case, we are also modeling the environment of the cell in terms of the substrates available and also the oxygen available for respiration. And what we have found in this case that indeed, if you enforce these trade-offs and this sort of resource allocation problem within an environment, you actually end up evolving two different strategies that can codepend onto each other. So one strategy is actually to increase your sugar uptake, but this causes an increased overflow of acetate in this case. And this increased overflow allows an acetate access to a different strategy to evolve, which uses this acetate and maintains a low glucose uptake. So this suggests that these kinds of interactions between cells through overflow of metabolites is possible under these kinds of trade-offs. We verified this experimentally using some experimental evolution experiments that others have done, but I'm not going to go into that. The second limitation is that I mentioned the presence of these redox reactions within the cell that allows this electron overflow. You can actually map those reactions to a reduction potential charge in terms of the half reactions involved. Okay? So you have different sugar conversions here, oxidations and reductions. Well, in this case, I'm showing all of them as reductions, but if you reverse a half reaction, obviously that becomes an oxidation. And you have a variety of such reactions within the cell, and then ultimately you have your terminal electron acceptors, which are at high reduction potentials. Within the cell, interestingly, many of these reactions are enabled by a set of so-called electron carriers, such as NAD, NADH, and ferredoxins. And these elements actually reduce and oxidize in between these reactions. But this picture is not as static as I'm depicting it here, but instead all these reduction potentials can change as the concentrations of the different compounds change. So this is a highly dynamic system, and it is possible that you're actually reaching thermodynamic bottlenecks as different reactions take place. And again, we found evidence that such thermodynamic bottlenecks happen and also linked to overflow reactions. So in this case, we're working with an organism that actually uses sulfate to respire. And in the absence of sulfate, this organism would produce or overflow hydrogen gas. But the overproduction of hydrogen gas is actually through a redox reaction, which is thermodynamically very close to zero. So this organism basically, in the absence of sulfate, will reach thermodynamic equilibrium and will cease the growth. The only way it can continue growing is actually by taking hydrogen away from the environment. And this is made possible in the environment by other bacteria or archaea that can actually live on this hydrogen. and then make it again thermodynamically feasible for this organism to grow. When we studied this system in detail, we found actually within the sulfate reducer, there are ways for it to invest membrane potential or ion gradients to make this hydrogen production possible. Again, bringing forth the idea that these thermodynamic inhibitions are quite real within the system and arise from imbalances in these redox reactions. Okay. So the final limitation that I like to highlight is that these redox reactions, and especially the final respiration process, can actually also generate radical species, which can then interact with other elements within the system, especially proteins, and damage those. So actually, while you're running this electron flow, you're also somehow toxifying yourself, and this hence creates a limitation to how fast you can run the electron flow. And interestingly, organisms has evolved ways to overcome this, and we found that in one particular ocean-dwelling bacteria, they actually produce manganese oxides that can then deal with these radical species, again, through redox reactions. So we're going, again, through redox reactions here, this time involving radical species. And the manganese oxide is interesting because it is a very strong oxidizing agent that can take care of these radicals. We are now also characterizing these manganese oxides with a view that they can involve in materials for batteries or other electrochemical applications. So I'm going to just finish the last few minutes with the consequences of this way of thinking about metabolism. So as I said, we see metabolism as an electron flow, and the electrons have to go somewhere through the system, and as soon as you create limitations in a variety of ways, these electrons have to be dealt with. And this creates interactions with, for example, transition metals or other cells through direct electron transfer, creation of electron-dense storage compounds or carrying compounds, or dumping of electrons onto organic acids or hydrogen, in which case you generate interactions between different cells. So it is an overarching view that we can potentially use to both study metabolism and to generate new ways of engineering metabolism. And we've been working on that front for the last few years, and I don't have much results here to show, but just to highlight the different directions in the last minute is that we can actually now interface with this redox metabolism through electrodes poised at specific potentials. And this has been already going on in biotechnology, but we are more interested in understanding the system and controlling the system in different ways. So in one organism, we are actually trying to replace the respiration with electrodes poised at different potentials, and we can show that in the absence of chosen respiring materials, strong electron acceptors, we can actually grow organisms on these electrodes and influence their metabolism. We are also trying to characterize these redox processes and their offshoots in the environment using scanning ion conductance measurements. These are basically nanoprobes that you can bring onto the surface of a cell and start measuring the redox reactions directly as they happen on and around the cell. And finally, we are trying to see whether species can directly transfer electrons without these intermediaries like hydrogen that I was explaining, to see whether we can sort of measure minute currents by sort of coupling a few numbers of cells together. And finally, we are also trying to model these coupled redox reactions in order to understand the temporal dynamics of the system. And what we are seeing is that these systems are very prone to generate bistability and oscillations, which are interesting nonlinear dynamics that we can potentially exploit if we can couple them with these kinds of electrode-based interferences. So maybe we can switch the system, if you wish, or make it start oscillate. And then the ultimate question is, if you want to try to do that, can we make that specific, and where we would want to put that kind of interaction within a given pathway or a metabolic system. So to finish off, basically, we're trying to apply this way of thinking about electron flows within a cell to try to explain all of physiological aspects that we see within a cell, as well as the cellular interactions in spatially organized systems. So I'd just like to highlight that we have established a bioelectrical engineering hub at Warwick where we try to sort of explore these ideas, not around metabolism, but also in broader contexts with a group of academics and also with some industrialists from the advisory board. So if you're interested in hearing about our events, please let me know. And with that, I'll just stop there and acknowledge some of the people in the group and the funders. Thank you for your time.
Any questions? I have a question.
Yes.
How would you characterize the rate of growth in this area, or it's a broad area of research, I guess, but
So, I mean, the conventional way for microbial cells is just to measure turbidity in a solution. But we are also using fluorescence microscopy, for example, to get growth rates of single cells, for example, and so on. But I'm not sure if you're trying to go somewhere with your question more specifically.
oh, my question was more kind of generally, are you seeing this type of research and looking at these reactions and potential applications of those reactions growing?
Oh, I see. So as a field, you meant.
Yeah.
Well, I hope so. I think there is already a well-established metabolic engineering field, and biotechnology uses a lot of these metabolic reactions to develop applications. What is new here is interfacing that with electrical systems and electrical ways of measuring things or influencing things. And that's definitely on the rise. I think more and more we realize many of the cells actually have capabilities to interact with electrodes, for example. Okay. Right. You opened the floodgates. Yeah.
So, do you actually believe in the word electrotrophic?
So you mean in the sense of, in what sense?
Feeding an organism with electrons from an artificial source.
From an electrode. Well, we are trying to do that. I think whether that happens naturally, there are some reports that that is happening naturally already. Not all the studies are very well controlled and so on. But I do think that that can be a possibility. Also, as you know, there are papers coming out now using mediators to interface rather than direct interfacing. I think that's quite promising. So yes, I think in the long run that will be possible. Yeah.
So, two things. Can you give me a sense of the time constant of these processes within a cell? About how long things take?
I mean, so metabolic reactions are on the scales of seconds, actually, to minutes. Now, when we're talking about cell division, etc., that can be in the scale of hours. But I think the level we want to work at is seconds to minutes. So we want to be able to influence metabolism. And then there will be a sort of knock-on effects on growth, etc.
Is there any tool that you have or use that allows you to get a potential gradient across the cell in real time? That's where I don't have a tool at the moment. That's where we're headed with the scanning ion conductance. That's why we want to use that technology to look at. So we're just mapping charge, for example, across cell, and we are hoping to be able to map pH, for example, over time, in real time on single cells. But those are challenging experiments.